take. Well, hi everyone. I am pleased that all the speakers before me talked about temperature because it will come into play for sure. So my name is Biplabendu. I go by Bilu, and I just finished my PhD in May. And I'm going to talk a little bit about what I was doing with Teresa de Becker at the intersection of chronobiology and behavioral plasticity in social insects, mostly ants, because I love ants. First and foremost, I want to thank all the people who have been very supportive throughout my PhD, and especially Teresa for introducing me to chronobiology, and of course, like Chelsea, Floria, and Caitlin for organizing this symposium. So thank you. So we all know life on Earth evolved on a planet that rotates, and it takes around 24 hours for it to complete one rotation, which creates some predictable daily fluctuations, right? Temperature being one of them, light being a big one, and all living organisms everywhere had to deal with a way to synchronize, adapt to this daily fluctuation. And the answer seems from studies done across all different fields is the biological clock. So I will talk briefly about what the clock actually comprises of, but let's talk about what the advantages are, right? The clock, once synchronized, can anticipate these daily fluctuations, and that means if you put yourself in a cave somewhere, you should be able to anticipate and drive this rhythmic behavior, which includes your sleep-wake cycles, your daily activity, metabolism, and physiology. The other and probably the more fascinating part about the clock is it is inherently plastic. And what it does is it means that the clock control rhythms can change with an organism's age, their physiology, but also their social environment. And let's talk about the classic example of plasticity, and that is the sleep-wake cycles. If you remember your teenage years, you do not want to wake up later than, I don't know, I still wake up at 10. It's mostly because you're not lazy, that's your clock, you're supposed to wake up later in the day, but as you age, you become older, you tend to wake up earlier and earlier. That is because the time at which the daily sleep-wake cycles work is changing and shifting with your age and your physiology. A similar but even more extreme case exists in all social insects that have been studied so far, is all social organisms that have a division of labor, where you have brood tending nurses and you have the foragers that are usually the older individuals, tend to show differences in their daily activity cycles. And what happens, at least in diachama, is when you isolate an individual and you put them in the presence of a brood, which is a larva, they tend to show no daily rhythms. They are working around the clock, pretty much shift work. But in the absence, in isolation, these individuals will show robust daily rhythms and activity, which is similar to what most animals do show. So there are multiple questions. How? How do they do something like this, where in humans we know like abruption of like the daily rhythms is associated with a lot of risks and even cancer. But more interestingly for me, what was fascinating when I joined my PhD was not much was known about the molecular basis of this plasticity. And it seems like it is linked tightly with the behavioral plasticity because even in queens, there is a shift in the daily activity rhythm when it is mated versus it's not mated. And in foragers versus nurses, every time they switch between these two behavioral groups as a response to social cues, not only the physiology changes in the gene expression, but also these daily activity cycles. So as I said, let's talk briefly about what the clock comprises of at the molecular level. And not much is known about the clocks in social insects. From what we know, it is very similar to mammals, and the mammalian clock is very similar to that of Drosophila and plants. And it's all comprises of a core oscillator, where you have a protein that rhythmically binds, a protein dimer, that rhythmically binds and expresses the repressor elements in a rhythmic manner, right? So that's why you get the oscillations. But turns out the activator complex also activates the expression of hundreds and thousands of other genes that gives you what we call the clock control genes, which in turn drives the rhythmic behavior and the physiology at not just the individual level, but also the collective level. So this is the background that I was in, and I was like, you know what, why not just look at all the genes in the ants and look at what actually is rhythmic, since we have some idea of what genes do in ants. So we set out to do this in the Florida carpenter ant, the Camponotus floridanus. Luckily, I was in Orlando. I could go in my backyard, collect ants, bring them back, and recreate, well, at least try to recreate the natural environment. So the first task was to identify the foragers and nurses in the colony. And what we used is what probably everyone is familiar with. You take two Tupperwares, you connect it with a big enough tube to kind of separate the nest from the foraging arena. And we kept the nest box in constant darkness while the foraging arena was exposed to 12 hours of light and 12 hours of darkness. And we used a GoPro to record and monitor the activity and the feeding of the colony. And this helped us not only just to monitor the activity, but also confirm that the colony was entrained to the light dark cycles. And once we were certain that they were, because these are nocturnal ants, and I'm not showing you the figure, but we quantified and it does look nocturnal, so you have to believe me or read the paper. So once we are all confirmed that this is entrained and we have identified foragers and nurses in the colony, and the definition of foragers is individuals that repeatedly go out foraging, so which we do identify using mark and recapture, and the nurses are individuals that are brood tending, so in direct contact with brood. We would then collect these individuals, 3 foragers and 3 nurses, every 2 hours over a 24-hour period. And once we have collected them, we dissect the brains and then we perform RNA-seq. So what we get at the end is a time course RNA-seq for both the different behavioral groups, which then allows us to look at daily rhythms in the gene expression. So the first thing we looked at is how many genes are oscillating every 24 hours. And what we find is more than, it's almost like 3 times as many genes that are oscillating every 24 hours in the forager brain as compared to the nurses. And this is similar to what was known in honeybees, because they did a microarray study like years before I started my PhD. But in our case, what we found is there's way more genes that are supposed to be oscillating because we used RNA-seq and we had a better depth and we had a higher resolution. But this also got us excited about the 700 odd genes that seems to be oscillating in both the groups, right? What are these genes, and also do they oscillate in the same manner? Is there any evidence of any plasticity? So we looked at gene expression, and what I'm showing you is a heat map when each row is a single gene and each column is a time point of collection, where the red tells you these are forager samples and the blue is the nurse samples. And the brighter yellow it is, the higher the expression. The darker it is, the lower the expression. And the first thing you notice is there's not much difference between how they oscillate between foragers and nurses. And when we looked for changes or differences in phase or amplitude, we did not find any significant difference, which tells us 2 things. First, the nurses that are seemingly arrhythmic and were considered not having a functional clock seems to have a lot of genes that are oscillating in a similar manner in both the behavioral groups, so there is something oscillating. And second, as you can see, the almost 3,000 genes that do not oscillate in the nurses might contribute to the fact that some of the behavioral output that we see, some of the behavior that we see is not cyclic anymore in the nurses as compared to the foragers. However, this still does not answer the question which I'm most fascinated with, is how does the switch between the foragers and nurses in the activity rhythms happen so fast? Because we all know like foragers and nurses can switch back and forth pretty fast. So we looked at the core clock. As I showed like earlier, there are a few components that are conserved across all animals, one of them being period gene. And this is the gene that is being expressed in a rhythmic manner in most animals. In foragers, it's 24-hour rhythmic, as you would expect. But in the nurses, in honeybees, it was previously reported not to be rhythmic. Now, they had a resolution of every 3 hours. They never really checked for lower frequencies, or sorry, higher frequencies. But we had the data, and this pattern seemed to be consistent in different genes. And at some point during COVID, I was like, you know what, why not just check it? So we checked, and lo and behold, we find that the period gene is not arrhythmic, rather it oscillates every 8 hours. Now, this got me excited. I wanted to be like, this is it, we found something. But we had to still check if this is a transcriptomic data set, so we have 10,000 genes, this is one gene. Is it a signal or is it something that is one-off? So we looked for other genes, and for sure what we found is we have more than 200 other genes that are involved in transcriptional regulation and protein phosphorylation, 2 processes that are required for a functional clock that seem to switch from oscillating 24 hours in the forager brains to oscillating every 8 hours in the nurse brains. And more excitingly, we also find GPCR signal transduction. Now, this is a process that's important for olfaction. And olfaction in ants and social insects is everywhere, right? It is involved in not just behavioral plasticity, but also their direct communication. So we know something's happening, and this is somewhere I think we need to like dive a little bit deeper into the roles of the 8-hour rhythms in ants. And I have something that I can discuss later if someone were to ask me why 8-hour rhythms. I will be happy to discuss. There's something in the literature about brood care, and I will go into that later. But I wanted to switch gears and talk about genes that we know drives behavioral plasticity in social insects. And two of the components that pop up everywhere is vitellogenin and juvenile hormone, right? So we wanted to look at two genes that regulate the titers of these two hormones. So these two proteins. One is vitellogenin, the gene itself, and then the other one is venom carboxylesterase 6, which is a juvenile hormone esterase. It degrades juvenile hormone. So we expected to see higher levels of expression of these two genes in the nurses as compared to foragers, and that's exactly what we see. The blue is for nurses and red for foragers. But what you also see is that this is not only differentially expressed, but also differentially rhythmic. For the first time, we find that vitellogenin itself is oscillating every 24 hours in the nurse brains. Now, this is bizarre because these are seemingly arrhythmic individuals that stay inside darkness chambers. So how? And the second question is why? Both of them, I do not have answers for you, but for sure, what this tells us is the story that the clock, the plasticity of the clock, and the plasticity of behavior in social insects, at least in our species, seems to be tightly linked. And this is pretty much what I wanted to discuss from the paper that just came out earlier. But I work on zombie ants, and in our lab, I did work on how the behavioral plasticity and the link between the clock might be hijacked by like a behavior manipulating parasite. And we do have similar patterns that emerge in that. But if you want to talk about that, I'm here. But just to recap, so I want you to take a few things home from this talk. One, that the brood tending nurse ants are not arrhythmic, at least at the molecular level. There are different set of genes that are oscillating that are functional for the clock. Second, there are several genes that are essential components of the clock and that can switch between oscillating diurnally versus oscillating in an ultradian manner. And third and last is genes that underlie behavioral plasticity actually seem to be under clock control. And they do show cost-associated differential rhythmicity. So if you want, this talk should be up, and the QR code should take you to an app that lets you explore all the different genes in the Floridanus genome. So it will tell you, at least if you're working on a single gene, if it oscillates or not, how does it oscillate. So, and you can find me on Twitter or reach me. So thank you for your time.
We have time for one question, if anyone has a question.
Yes.
I was to ask you, are the 8-hour?
So, can you repeat your question? If I understand.
So I was wondering if you could speak near the mic,
Oh,
that way
sorry.
people can hear you.
Yes. Sorry.
I was wondering if the nurses have all the same 8-hour cycle, or if they're all shifted at different times, if they don't have any external cues.
So it seems like, so what we did is,
Repeat your question into the mic.
yes. So she asked if the nurse ants, if the 8-hour rhythms that we see, is it shifted for different nurses or is it the same? So sadly for us, we pulled all the samples. So what we have is one line for nurses and one line for foragers. But what we do see is the different genes that seem to switch are switching in exactly the same phase, similar to period. So it's not that these genes are slightly shifted, they're all oscillating in the same manner, meaning that there is some control, some activator that is activating these genes to oscillate every 8 hours. Does that answer your question? Yeah. Okay, thanks.
All right, thank you for that, Hilu. And so sorry about that.